Hello everyone. So in this video we are going to talk about chromatography, which is an important topic for the unit 13 methods of biology if you are preparing for CSR UGC NET JRF, right? So this is the part 1 of chromatography, this video. So let's see what it will cover. So the contents or the important topics that we need to cover in chromatography is its introduction, what is chromatography, classification of chromatographic methods, adsorption and partition chromatography, and there are rest which we'll cover in the second video, while in this video we'll only cover up to absorption and partition chromatography. So let's start with that. Chromatography introduction, right? So chromatography is based on a very simple principle, which is that the sample to be examined, called solute or analyte, is allowed to interact with the two immiscible phases, a mobile phase and a stationary phase, right? So when there for example we have a compound, for example we have chlorophyll. Now chlorophyll contains many compounds within it: chlorophyll A, chlorophyll B, chlorophyll C, carotenoids, right? So chlorophyll as a whole consist of many other compounds like chlorophyll A, B, C, carotenoids, all put into a mixture that is chlorophyll. Now this chlorophyll, we may need to separate these different compounds from the chlorophyll mixture, right? We may need to separate chlorophyll A from the rest of the compound, or we may need to separate chlorophyll B or the carotenoid from the rest of these compounds, right? So when we need to do that, uh, we use chromatography as a technique to separate different compounds from a mixture of a compound, right? So what we need to do is the solute or the analyte from which we need to separate different things is allowed to interact with two immiscible phases. Immiscible means two things that cannot be mixed or which do not mix. For example, oil and water. These two things are immiscible, they do not mix, right? So there are two immiscible phases, a mobile phase and a stationary phase. So to separate different compounds from a mixture of compound, what we need to do is we need to have a mobile phase and a stationary phase. Now as the name suggests, the mobile phase will be the phase that can move, and the stationary phase will remain stationary, or this is the phase that would not move, right? So this is what we require to separate different molecules from a mixture. The two immiscible phases could be a solid and a liquid, or a gas and a liquid, or a liquid and another liquid, right? So it could be the solid and a liquid, which means that the stationary phase will be solid and the mobile phase will be liquid, or it could be gas and liquid where mobile phase is gas and liquid is the stationary phase, or it could be liquid where both the stationary and the mobile phase are both liquid. The stationary phase, which may be solid or liquid, does not move, right? The stationary phase does not move, while the mobile phase moves the sample through the stationary phase, right? The mobile phase moves the molecules, while the stationary phase remains stationary. If the mobile phase is liquid, it's liquid chromatography. If the mobile phase is solid, then it's called solid chromatography. So the naming structure is based on the mobile phase, right? If we call it liquid chromatography, liquid chromatography would mean that the mobile phase is liquid. So the naming structure always depends on the mobile phase. If it's liquid chromatography, it means that the mobile phase is liquid. If it's solid chromatography, it means that, sorry, if it's solid chromatography, it means that the mobile phase is solid, right? If the mobile phase is solid, then it's called solid chromatography. The same thing goes for gas. If the mobile phase is gas, then it's gas chromatography. The two phases are chosen so that the components of the sample distribute themselves between the mobile and stationary phases to varying degrees. This means that, for example, take a mixture of molecules, right? We take a mixture of molecules and we need to separate these different compounds from this mixture. Now we put it through, there is a solid phase and there is a liquid phase, right? Now some of these molecules in the mixture will be more soluble in the mobile phase, which will move, and some of these molecules may be more soluble in the stationary phase, right? If it is a mixture of molecules, some of these molecules might be more soluble in the mobile phase and some of these molecules may be more soluble in the stationary phase, right? So if those molecules that are more soluble in the mobile phase will move through the column and will elute out, will come out of the column first, right? We'll get them first. While those, because mobile phase moves, it will move through the column or the jar or whatever in which you are carrying out the chromatography. It will move because it is more soluble in mobile phase, and mobile phase moves. So those molecules of the mixture which will be more soluble to the mobile phase will move out first, and we will get them or will be eluted out first from the column. While on the other hand, those molecules which are more soluble in the stationary phase is that phase. So if it is more soluble in the stationary phase which do not move, it will elute out later. It will come out later. Right. So that is about chromatography. Let's move forward. It was the introduction. So here you can see this is paper chromatography. This figure shows paper chromatography. In paper chromatography, the stationary phase, the phase that does not move, stationary phase is the paper, while the mobile phase that moves is some organic compounds. Here you can see the liquid, right? The liquid which generally contains ethanol and other organic molecules, right? So you can see the paper is the solid or the stationary phase. It's a kind of, and this is the liquid and the mobile phase, right? So the mobile phase moves. As you can see, if we this is we are separating the mixture of chlorophyll, right? So if we put chlorophyll here, somewhere here, right? If we put chlorophyll, a pigment of chlorophyll right here, and we allow the mobile phase to move upward through the paper, through the stationary phase, the paper will not move, but the mobile phase, which contains some organic liquids, will move through the paper or the stationary phase, right? And those molecules of the chlorophyll which are soluble in the mobile phase, will move along the paper and will be separated out according to their RF values, right? If it is more soluble in the mobile phase, we'll find it at the top, while if it is less soluble in the mobile phase, we'll find it at the lower half of the solid state, right? If it's it has more affinity towards the solid phase of the paper, then we'll find it at the lower end of the paper, right? So let's see what happens. The carotene is found here, the xanthophyll is found here, the chlorophyll A and chlorophyll B, right? So the chlorophyll A and chlorophyll B, you can say that they have more affinity towards the stationary phase than the mobile phase. While the carotene, which has the highest RF value, right, it has moved the furthest from the base with the mobile phase, has more affinity towards the mobile phase, right? So that is about that. Let's move forward. So the classification of chromatographic methods. So there are many chromatographic methods, and they can be classified in three fundamental ways. The first is based on the shape of the chromatographic bed, right? The second is based on the physical state of the mobile and stationary phase, and the third is based on the mechanism of the separation. So let's see their details. So based on the shape of the chromatographic bed. So based on the shape of the chromatographic bed, there are two types of chromatography. The first is planar chromatography and the second is column chromatography. So based on whether the solid state or the chamber where you are carrying out the chromatography is planar or is columnar, there are two types of chromatography, right? If you are carrying out the separation in a column, you can see like this cylinder kind of structure which is called column. If you are carrying out the separation in something like this, then it's a columnar chromatography. And if you are carrying out it in some kind of planar, like uh, uh, aluminum plate or what we saw earlier, the paper chromatography or the TLC plate, whatever that is, if it's a planar structure, if it's a 2D structure, then that is paper chromatography or the planar chromatography, right? And if it's carried out in a column, then it's columnar chromatography. So let's see their details. In planar chromatography, the stationary phase is spread on a flat planar surface, right? The plane can be paper impregnated by a substrate acting as a stationary phase, paper chromatography, or a thin layer of substance acting as a stationary phase spread on a glass, metal, or plastic plate, which is thin layer chromatography. So you can see both. Here you can see that this is the paper chromatography where the stationary phase is a paper, right? A planar surface, because it's a kind of a planar chromatography. The stationary phase is paper and the mobile phase are the liquids at the bottom, right? So this is a kind of planar chromatography. On the other hand, there is also another kind of planar chromatography, which is the TLC, the thin layer chromatography. In thin layer chromatography, there is a glass plate put into a jar, which is the chromatographic chamber, right? We put the glass, which is absorbent coated, and the absorbent could be alumina or the silica, something like that, right? Silica gel, which could be put upon the glass plate as the stationary phase, and then we put the sample or the analyte on that glass plate, and then we put the solvent, or which is the mobile phase, at the bottom, right, at the bottom of the chamber, and we allow the different components of the mixture to be separated based on if it has more affinity towards the solid phase or the stationary phase, or if it has more affinity towards the liquid or the mobile phase. Now if the component will have more affinity towards the mobile phase, it will move faster, and if it will have more affinity towards the solid phase or the stationary phase, it will move slower. So this is the TLC, thin layer chromatography, right? And this is the paper chromatography. Both are planar chromatography, right? So in thin layer chromatography, the stationary phase is thin layer of silica gel or aluminium on a glass or metal plastic plate. So as you can see, this is a metal or plastic plate which is put inside a jar or the chromatographic chamber, and this glass plate is covered with a layer of silica gel or alumina, right, which act as the stationary phase. This silica gel or alumina act as the stationary phase, which is present on the glass plate. Now let's move on to the next slide. The next we saw the planar chromatography. Let's see the column chromatography. The stationary bed is within a tube, right? As the name suggests, column. It needs a column, right? It is a cylinder kind of structure. It has a cylinder kind of structure, and within the cylinder there is a packing material, right? Something like this, right? It's the column is packed with different molecules, right? Um, it it has a packed column and concentrated inside the tube wall, leaving an open unrestricted path for the mobile phase in the middle part of the tube, right? So it is packed column and the mobile phase move through these spaces, while these molecules are the stationary phase, while the mobile phase move through these gaps, right? So that is column chromatography. As you can see that this is a column, and you have loaded the sample right here, and this is the whole stationary phase, and there is now mobile phase, right? Now if the sample you have loaded has strong interaction with the um, stationary phase, then it is moving slower, while if it has weaker interaction with the stationary phase, then it's moving faster, right? So you can see it's moving faster. So the molecules which elutes first will come out first, has lower affinity with the stationary phase and has more affinity with the mobile phase that is moving. This is now column chromatography. Now let's move on to chromatography based on the physical state of the mobile and stationary phases. Now, based on the physical nature of mobile phase, there are two types of chromatography: gas chromatography and liquid chromatography. Now, the chromatographic name is always given on based on the mobile phase, right? In any chromatography, if the mobile phase is gas, then it's gas chromatography. If mobile phase is liquid, it's liquid chromatography, right? So it's always based on whether the mobile phase is gas or liquid. It will be named either gas chromatography or the liquid chromatography based on the mobile phase, physical nature of the mobile phase. Now further, based on the physical nature of the stationary phase, there are two subgroups of gas chromatography, which is gas solid chromatography and gas liquid chromatography, right? So if we talk about gas solid chromatography, it would mean that the mobile phase is gas and the solid is the stationary phase. So the first word that you will find is always the mobile phase, and the second is the stationary phase. So if it's named gas solid chromatography, it means that the mobile phase is gas and the stationary phase is solid. And if it's named gas liquid chromatography, it would mean that the mobile phase is gas and stationary phase is liquid, right? Next, if it is named liquid solid chromatography, it means that mobile phase is liquid and stationary phase is solid. If it's liquid chromatography, it means both mobile and stationary phase are liquid. Both are liquid. Let's move on to the next. So now it's based on the mechanism of separation. So based on the mechanism of separation, the chromatographic techniques use several types of mechanism to separate analytes. It could be partition, adsorption, size exclusion, affinity, and ion exchange chromatography. Right, so let's see all of them in detail. Adsorption and partition chromatography. So, chromatographic techniques can be classified into two main categories, partition and adsorption chromatography, right? Depending on how the solute interact with the stationary phase. If there is adsorption between the stationary phase and the molecule, then it's adsorption chromatography. If you can find two immiscible liquids and it depends on the solubility, then it is the partition chromatography. If the separation is based on solubility between two kind of liquids, then it's partition chromatography, right? So the mode of interaction between the sample components and the two phases can be classified into two types,